Let us look at lymphocyte development, the way that lymphocytes develop in the body. The recognition of antigen by the immune system occurs using 4 different types of molecule. The pattern recognition receptors, which are present on cell surfaces, inside cells, and secreted or released from cells, recognize pathogen-associated molecular patterns and damage-associated molecular patterns. We have maybe a small few hundred of these. There are new ones being discovered all the time. Let's say 100 or 200 or so different pattern recognition receptors that we each possess. Each of these is individually encoded by a gene. So for each pattern recognition receptor, we have a different gene, and they are not very variable from one individual to another. So for a given pattern recognition receptor, probably your pattern recognition receptor is exactly the same as mine. Let's say TLR4. Probably your TLR4 is exactly the same as mine. In other words, they have low polymorphism at the genetic level. They don't vary from one individual to another for each individual pattern recognition receptor. Then we have the major histocompatibility complex molecules. These are cell surface molecules, and each different MHC variant can bind many different peptides derived by processing of protein antigens. And in each individual, there are around about 12 different MHC molecules with respect to what we call the classical MHC molecules, the main MHC molecules. We each have about 12 of these. Each protein is individually encoded. So for MHC class 1 in the human, we have HLA-A, HLA-B, HLA-C. For class 2, we have HLA-DP, DQ, DR. And each of those will have its own gene. But at each gene locus, let's say the HLA-A gene locus, the sequence varies hugely from one individual to another. So my HLA-A molecules are probably different to your HLA-A molecules. And therefore, they are described as being extremely polymorphic between one individual and another. Then, at the level of the cell surface of lymphocytes, on T cells, we have the T cell receptor. This molecule is highly specific for a given combination of a peptide that is produced by processing a protein antigen put together with MHC molecules. So T cell receptors are highly peptide MHC specific. And we can create millions and millions and millions of different T cell receptors. Each of us can do that. Quite remarkable. And this is due to a unique genetic recombination mechanism to create diversity. The B cells have a similar molecule on their surface. It's a different molecule encoded by different genes, but in many ways it's similar. But on the B cell surface, you have a B cell receptor, or BCR, but this molecule, unlike the T cell receptor, can also be released from B lymphocytes. So it's produced in both a cell surface and a secreted form, whereas the T cell receptor is only ever produced in a cell surface form. Antibody recognizes antigen in a highly antigen-specific way. Just like we can produce millions of T cell receptors, so we can produce many millions of B cell receptors and antibodies. Okay, the B cell receptor is simply a transmembrane form on the cell surface of a B cell. And we can do this just like we can for the T cell receptors because of this unique genetic recombination mechanism. So we have a bunch of T cell receptor genes, we have a separate bunch of B cell receptor genes or antibody genes, and these can recombine by a mechanism that's completely unique. There's nothing else in the body does anything like this. From a small number of genes, a relatively small number of genes, we can make millions and millions and millions of different antigen receptors. And it's one of the things that makes the adaptive immune response quite so remarkable. So B cells, as their name suggests, B, develop in the bone marrow. So the bone marrow is where B cells develop. And in fact, virtually all the cells of the immune system start off from multipotent hematopoietic stem cells in the bone marrow. And B cells can become fully mature within the bone marrow. They don't need to go anywhere else to become mature B cells. They develop within the bone marrow. Once they are mature, they can leave the bone marrow and travel to the secondary lymphoid tissues, the mucosa-associated lymphoid tissue, the lymph nodes, and the spleen. So let's have a look at how B cell development in the bone marrow occurs and the stages in B cell maturation in the bone marrow. I've already mentioned they start off from hematopoietic stem cells. These develop into pro-B cells, those hematopoietic stem cells that instead of becoming a mast cell or instead of becoming a dendritic cell, those that are going to become B cells initially become pro-B cells. The next stage in their development is to become a pre-B cell. Then they develop into immature B cells and, of course, eventually into mature B cells. There are two stages in this development process where these B cells undergo very, very extensive cell division, in other words, proliferation. That's at a very early stage in their maturation, at the stem cell pro-B cell stage, and then later on, at the pre-B immature B cell stage, they undergo further massive rounds of proliferation. They're dividing all the time, but these two stages in their development are characterized by particularly extensive cell proliferation. There are two genes that are called RAG1 and RAG2. It stands for recombination activating genes. I've already mentioned that the immunoglobulin genes in the B cell undergo recombination to create diversity. These recombination activating genes obviously play a role in that, as their name suggests. And these genes are also expressed at two different points in the development and maturation of B cells, at the pro-B, pre-B stage, and then they can become re-expressed later on in immature B cells to fine-tune the antigen receptor on the B cell. There's another molecule which we'll hear about shortly called TDT, terminal deoxynucleotidal transferase. Bit of a mouthful, that, isn't it? We normally just refer to it as TDT because terminal deoxynucleotidal transferase is quite a long word to say. So TDT. And that's expressed again at the stage between pro-B cells and pre-B cells. If we look at the immunoglobulin gene DNA in the B cell and the RNA that's produced from that DNA, in the stem cell stage and the pro-B cell stage, these immunoglobulin genes are what we call in the germline configuration. That means how these genes are inherited through the sperm and the egg, the germline, and every single cell in the body will have this DNA, just like you have genes for all the different genes you have in all the different cells. And in all the cells in the body, they will be in the germline orientation or configuration. And it's only once cells develop to become B cells that these immunoglobulin genes recombine. And this recombination initially starts off at the pre-B cell stage, and some segments within the heavy chain genes for the immunoglobulins, because immunoglobulins consists of a heavy chain and a light chain. The first event that says, that cell's going to become a B cell, is when the B cell recombines its heavy chain genes. And it recombines 3 gene segments, as we'll see shortly. They're called V, D, and J, variable diversity and joining. Once that recombination process has occurred in the pre-B cell, messenger RNA is produced for the heavy chain, and that includes a segment that is from the constant region called mu. So you get mu mRNA in the pre-B cell. As that cell then develops to the final stage before full maturity, in other words, the immature B cell, the recombined heavy chain gene associates, the product of that gene associates with the product of a recombined light chain gene. And in the light chains, the V gene segments and J gene segments that recombine produce a light chain protein. So now we have Ig for the DNA that's recombined in the heavy chain and the light chain. We have a mu heavy chain protein and either a kappa or lambda light chain protein produced from the kappa or lambda messenger RNA. And then finally, we end up via alternative splicing of the V, D, and J with a constant region of the IgM antibody, which is encoded by a C mu gene, or the IgD antibody encoded by a C delta gene. So the stage of maturation, early on, the immunoglobulin genes are not recombined, so there's no expression. In order to express these genes, they need to be recombined. They only recombine in B cells, and they only recombine at the pre-B cell stage. So you start off at the pre-B cell stage with the first indication that this cell is going to become a B cell. It has cytoplasmic mu heavy chain, and on the surface it has what is called the pre-B cell receptor, which is a surrogate light chain. It's not the proper light chain, but it's a sort of intermediate light chain that's produced to put together with the mu heavy chain. And then once the light chain genes have been recombined, you get the full mature antibody molecule on the surface of this immature B cell. So it's a mature antibody, but it's on the surface of an immature B cell, and this is membrane IgM. And then, as well as IgM, another class of antibody, the IgD class, is put on the surface of the mature B cell. As well as having antibodies on their cell surface, B cells have thousands of other molecules on their cell surface, just like every cell does. And we can use certain molecules on the surface of B cells to identify at which stage in maturation they are. So, for example, the molecule CD34 is present on the surface of the stem cells. It's also present on the surface of the pro-B cell. But additional molecules become expressed, such as CD19 and CD10. And then as those pro-B cells develop into pre-B cells, there is low expression of a molecule called B220. But as that pre-B cell develops into an immature B cell, the expression of CD34 is lost. So if a cell has a molecule on its surface, we describe it as a CD whatever positive cell. If it doesn't have expression, we describe it as a CD whatever negative cell. So, in other words, the stem cells, the pro-B cells, and the pre-B cells are CD34 positive, whereas the immature B cell is CD34 negative. At that stage of the immature B cell, there is a low-level expression of IgM antibodies on the surface of the B cell. And then as the B cell further matures to become a fully mature B cell, the level of expression of IgM increases. So where are these different types of B cell, these different stages of maturation found? Well, the stem cells, the pro-B cells, the pre-B cells are all within the bone marrow. And then as these cells develop into immature B cells, they can leave the bone marrow and enter what immunologists refer to as the periphery. Rather strange term, that, the periphery. Simply means anywhere outside of the primary lymphoid tissues. So anywhere in the body that isn't the bone marrow or the thymus, immunologists refer to as the periphery. And the mature B cells are found in the periphery, or move to the periphery. The response to antigen, clearly if the B cell doesn't have an antigen receptor on its surface, it can't respond. It has no way of responding to antigen. So because there is no surface antibody on stem cells, pro-B cells, or pre-B cells, there's no response to antigen. At the immature B cell stage, when there is low-level expression of IgM, the B cells can undergo what is called negative selection or deletion. And this refers to the fact that if those B cells encounter molecules from our own body, what we call self-antigen, then they become deleted. Because we don't want B cells that can attack our own body components. We want B cells that can fight infection coming from outside the body. So there'll be deletion of B cells that recognize self-antigen, and there can be receptor editing. And this is why RAG1 and RAG2 get re-expressed at this stage, so that the receptor can be changed. If it reacts with self, it can be changed to a different receptor that reacts with a foreign pathogen rather than reacting with self. And then once they're mature, of course, the B cells are fully able to respond to antigen. I mean, that's how it should be. Obviously, you need the B cells to be able to respond. And they can become activated, they can proliferate, divide up in number, and they can differentiate and become specialized, particularly become specialized to produce a progeny of the B cell that is called a plasma cell. And what plasma cells do is they secrete very large amounts of antibody of the same specificity that acts as the B cell receptor on the surface of the B cell. So in the bone marrow, we have these pre-B cells, this early stage of development, and they have on their cell surface not a mature form of the antibody molecule, but a preliminary version, if you like, of the antibody molecule, where you have the normal version of the IgM heavy chain, which is called mu heavy chain, but it's not a mature version of the light chain. They have what we refer to as a surrogate light chain, and this is made up of two components. One is called V pre-B, and the other is called lambda 5. And then as the pre-B cell matures into an immature B cell, it swaps the surrogate light chain for a real light chain. And that can either be the kappa light chain or the lambda light chain. So that the mature B cell now has on its cell surface a proper mature version of the antibody molecule. And initially, these mature B cells that have not yet encountered antigen, we call them naive B cells, they've not yet met antigen at all, they express on their cell surface two different classes of antibody, IgM, surface IgM, and surface IgD. And these cells have left the bone marrow and traveled to the secondary lymphoid tissue. And they sit in wait in the secondary lymphoid tissue in case their particular antigen, the antigen they recognize, comes along. We heard a few seconds ago how from a relatively small number of genes, you can make millions and millions and millions of different antibody molecules and millions and millions and millions of different T cell receptor molecules. So let's have a look at how that works. This is the immunoglobulin heavy chain gene locus. Happens to be on chromosome 14. Doesn't really matter where it is, but happens to be on chromosome 14. And it consists of a number of gene segments, they're sometimes called mini genes. And essentially, there are 4 bunches of gene segments for the immunoglobulin heavy chains. It's a bunch of segments shown in blue here that are called the V or variable region gene segments. And we have about 40 of these. So one's called VH1, VH2, VH3, VH4, and so forth. Around about 40 in total. We then have a bunch of D segments, diversity segments, shown in green here, and we have about 27 or so of those. Downstream, or 3' in the DNA sequence from the D segments, there's a bunch of J segments. We have about half a dozen of those, indicated in red here. And then further downstream, we have a total of 9 constant region gene segments. Just one is shown here, C mu, but there are a total of 9. So on the heavy chain gene locus, 4 bunches of gene segments, V variable, D diversity, J joining, and C constant. The light chains are organized in a somewhat similar way. The organization isn't exactly identical, but essentially there are bunches of gene segments. And the main difference really is that in the light chains, there are no diversity gene segments. So it consists simply of a bunch of V segments, J segments, and C segments without any diversity segments. So upstream, just like in the heavy chain, upstream of each variable sequence is a leader sequence that takes the protein to the cell surface when it's been translated. And for the light chains, we have two different versions, kappa and lambda. This is lambda shown here. Again, happens to be on chromosome 22. That's not really that important unless you're obsessed with which chromosomes particular genes are on. But the important point is there are a bunch of V gene segments, not just one, but several, and around about 30 V lambda gene segments. And then the way that the joining segments and the constant segments is organized is a little bit different to in the heavy chain. But as you can see, there are a number of J segments and a number of constant segments. And if we were to look at the kappa light chain, the other variant of the light chain, again, it's on a different chromosome. So the heavy chain's on one chromosome, kappa light chain on another chromosome, lambda light chain on another chromosome. But the arrangement is similar. Bunch of gene segments. Remember, in the light chain, no D segments. So heavy chain, VDJ, light chain, just V and J, and then together with the constant segments. So for the kappa light chain, we have about 40 variable, 5 or 6 or so joining. It varies from one individual to another, but that's sort of typical number would be 5 joining segments for kappa and then constant region, as you can see. So let's now look at how the immunoglobulin genes recombine in B cells. What you can see in front of you are the immunoglobulin heavy chain genes in what is called the germline configuration. And these will be present in this configuration, in nerve cells, in liver cells, in all the different cells in the body. And it's only in the B lymphocyte that these genes will recombine in order to generate an antigen receptor. And in each individual B cell, the B cell will choose a different combination of V, D, and J gene segments in order to create a different B cell receptor. So you may have one B cell that recognizes Streptococcus, for example. A different B cell might recognize Staphylococcus, another B cell Candida, and so forth. And it needs a different antigen receptor to recognize each different antigen in a specific way. So to take this B cell for an example, this B cell has decided to use the D segment D2 and has placed it next to the J segment J4. That's the first stage. The second stage is to select one out of the 40 variable segments and place it next to the already recombined D and J segment. So this B cell decides to use V4 and places it next to the already recombined D2 and J4. So in this B cell, you'll have a continuous stretch of DNA that is V4, D2, J4. That DNA is then transcribed into the primary RNA transcript, and that is processed into messenger RNA. And that primary transcript includes the first 2 constant region genes, C-mu, that will encode an IgM antibody, and C-delta, that will encode an IgD antibody. And by alternative splicing of the primary RNA transcript, you produce messenger RNA that either will encode a IgM antibody using C-mu, as in this particular example here, or alternatively using C-delta to encode an IgD antibody. And the important point is the same V, D, and J sequence is being used for both of those classes of antibody. So both the IgM antibody and the IgD antibody in a single B cell will be of identical specificity. So if the IgM is against Staphylococcus, the IgD will be against Staphylococcus. That messenger RNA is then translated into protein, and the constant region gene, as the name suggests, encodes the constant region of the antibody molecule, in this case an IgM antibody, and the variable region is made up of 3 different gene segments: the V segment, the D segment, and the J segment. So although the variable region in the protein is just referred to as the variable region, it's actually encoded by 3 different gene segments. Not only the variable gene segment, but also the diversity and the joining gene segments. Within the variable region of antibodies, there are 3 regions that are hypervariable. They vary even more between one antibody and another. And these 3 hypervariable regions are referred to as the CDRs. That stands for complementarity determining regions. So we have CDR1, CDR2, and CDR3. And as you can see from this illustration, CDR1 and CDR2 are encoded entirely by the variable gene segment, whereas CDR3 is encoded by a combination of V, D, and J gene segments. What that means is that CDR3 is much more variable because it's the combination of 1 out of 40 variable, 1 out of 27 diversity, 1 out of 6J. Whereas at least initially, the CDR1 and CDR2 are not nearly as diverse because they can only be selected from 1 out of 40 variable gene segments. So the antibody is then produced. This is the heavy chain you see here. The light chains undergo a similar recombination process, but remember there are no D segments in the light chains, so it's just a recombination of 1 out of a number of V segments being placed next to 1 out of a number of J segments. So on this particular diagram, we can see how the CDR1, CDR2, and CDR3 hypervariable regions sit towards the tip of the antibody and are present in both the heavy chain and the light chain. So the variable region of the light chain and the variable region of the heavy chain are next to each other, and then below that is the constant region of the heavy chain and the light chain. Let's take a few moments to look in a little bit more detail at how this genetic recombination process actually is mediated. It really depends on two things, or requires two things. It requires a number of different enzymes that catalyze the recombination process, but it also requires recognition sequences for these enzymes. In other words, substrates for these enzymes that are within the DNA sequence. Okay? They're not in the part of the DNA that encodes the protein, but they lie immediately adjacent to that part. And they're called recombination signal sequences, or RSS. And the enzymes that recognize these recombination signal sequences, they're nucleotide sequences, as I say, flanking the V, D, and J segments. The enzymes that recognize, or the two most important enzymes that recognize these recombination signal sequences, are called RAG1 and RAG2. So heavy chain VDJ recombination, and likewise light chain VJ recombination, is mediated by the recombinase enzymes RAG1 and RAG2, stands for recombination activating genes, even though they're actually proteins that come from those genes, and they recognize these recombination signal sequences, or RSS, which are nucleotides flanking the V, D, and J gene segments. And as you can see from this particular picture, the V segment has an RSS immediately 3' or downstream of the protein coding sequence. The D segment has two RSSs, one either side, and the J segment has a single RSS 5' or immediately upstream of its sequence. So following successful heavy chain recombination of VDJ, the light chain is then recombined, because an antibody needs both a heavy chain and a light chain. And here we can see the variable region of the kappa light chain, around about 40 different variable segments for the light chain in the for kappa light chain, and 5 or so joining gene segments. And this particular B cell has decided to recombine its kappa light chain using V kappa 2 and recombining it next to J kappa 2. And then, just as we saw for the heavy chain, it will make a primary RNA transcript, it will process that into message, and then translate that into protein. And again, the CDR3 in the light chain is the most variable because it's a combination of any one out of those 40 Vs put next to one of those 5 different J gene segments. So the recombination occurs in both the heavy chain and the light chain. And that recombination, being a fairly random process, is not actually always successful. Sometimes it doesn't work. So there might be an out of reading frame sequence generated, or maybe a stop codon is generated. So in some ways it's a bit of a wasteful process, given that it doesn't always work, but it's an incredibly powerful process because it means that from a handful of genes, you can make millions and millions and millions and millions of different antibody molecules, and likewise many, many million different T-cell receptor molecules. So the sequence of events is that there are 2 attempts at recombining the heavy chain. Remember, we have 2 copies of each chromosome. So you've inherited 1 copy of your immunoglobulin heavy chain genes from your mom and another copy from your dad. And given that this random process is not always successful, maybe you fail to make, a B cell fails to make a productive, what we call productive recombination on one of the chromosomes. All's not lost, it can try the other chromosome. As long as 1 attempt is successful, then it will go on, the B cell will go on and recombine the light chain genes. It starts off with the kappa light chain. Again, there are 2 attempts because you have 2 versions, one you've inherited from your mom, one from your dad. And in fact, for the light chains, there's a total of 4 possible attempts because as well as having the kappa light chain genes, as we've already heard, we have the lambda light chain genes. So if both recombinations on the kappa chain are unsuccessful, there's still hope left. There's still a chance because there are 2 lambda chain gene loci. So hopefully the B cell will be successful in both the heavy chain recombination, 1 out of 2 choices, light chain recombination, 1 out of 4 choices, and produce a functional antibody molecule that will then be put on the cell surface. It's important that B cells are specific for 1 single antigen. So for example, you'll have 1 B cell that's specific for Staphylococcus, another for Streptococcus, another for Candida, another for cytomegalovirus, and so on. And in order to ensure that the B cell is only specific for a single antigen, you need to just have 1 heavy chain sequence and 1 light chain sequence. And that is ensured by a process that is referred to as allelic exclusion. The result of allelic exclusion is that if the first attempt at recombining the heavy chain is successful, recombination on the other chain is prevented. It's not allowed. So as the first recombination is successful, you make a protein, there's a feedback mechanism that prevents recombination on the other heavy chain locus. Likewise, for the light chain recombination, if there's a successful recombination of the light chain, either the kappa light chain or the lambda light chain, it prevents subsequent recombination of other light chain loci. So allelic exclusion ensures that there isn't a mixture of different heavy chains or different light chains for a given B cell. Just 1 heavy chain, 1 light chain. There'll be millions of copies, but they'll all be identical.